professor, physician, epidemiologist. I am Dr. Shree Banerjee. Now, looking into this primer a little bit more, one of the features that I think it's important to include within the context of this video is providing what types of tools are available in each category. So, what I like about this classification system is that under each category there's several programs listed. So Plink, for instance, is listed under quality control and association. So the Rico Pili is there. And then imputation, there's some categories. Association. So Plink is one of the largest, and I've actually worked with this in the past, Plink 2. But then if you're looking to do statistical fine mapping or meta-analysis, variant annotation, enrichment or gene set analysis, QTL analysis, genetic correlations, causality, PRS analysis, TWAS, similar to GWAS, but it's transcriptome-wide association studies. So then you look for different software, and so these are all available within R. These are software packages which are very much in demand and provides good insight in all of these studies. Now, there's a phenomenon that the effect size of newly discovered alleles tend to be overestimated, which is true. So then you have to then kind of take a look at additional studies. And then another note that is on the left is odds ratio, an effect size estimate of a risk factor that quantifies the increased odds of having the disease per risk allele count in genome-wide association studies, or one standard deviation increase of the polygenic risk score. So that's how odds ratio is defined. So these are good definitions. Again, it's a primer, so it has some good learning points as you're moving along, and they go through various scenarios and types of statistics that you want to be familiar with. Now, this is not as recipe oriented as you would see a set of GitHub instructions. That is a lot more step 1, step 2, much more recipe oriented. This is much more descriptive, but at least you'll get a lot more of the theory behind this and definitions. Be on the lookout for definitions on the left. For instance, the transmission disequilibrium test is a family-based genetic association test in which alleles transmitted to affected offspring are contrasted with alleles not transmitted. These are the different types of tests that you can kind of run. A statistical technique that's usually run is rare variant burden testing, and that is where the number of rare alleles per gene is used to determine genetic association with the trait. So then there's the imputation workflow in box 2, which is really interesting. Goes into the imputation of ungenotyped single nucleotide polymorphisms. And so this can be done through online imputation servers. So this provides you with all the necessary steps, and the statistical fine mapping becomes interesting. They actually, one of my favorites in this paper also is the fact that in Table 2, they provide you not only with the list of the biobanks that are available all over the world, but they also provided kind of the ancestries that are covered within the biobank data sets. And I personally am a member of the UK Biobank, and there's also several places where you can um, receive genetic information and start your own analysis. It's a matter of finding genetic information. And these are several repositories which are readily available, which you can kind of. So the process for the UK Biobank, the approval process, requires you to submit a resume and several of your publications, things like that. So different places have different sorts of uh, requirements to try to join and be a part of collaboration. Um, I talked about the All of Us initiative in another one of my videos, so I encourage you to look into that. It's interesting. It's an NIH initiative, and many of you might have heard of 23andMe and the research arm of 23andMe, and that also is interesting. There's a postdoc program with 23andMe, and so and also they have 23andMe COVID-19 study and a data set um, that pertains to the COVID-19 study, um, which would be something that would be interesting as well, um, considering that genetic information is not necessarily always available. Um, so I was looking into some of the information that's included in here for this. So this would be something that's interesting, which and unique in that this is um, actually connected to a corporation, and you potentially have access to genetic information from there. So, bottom line, if you're looking for research in genetic epidemiology, trying to understand the effect, the phenotypic effect, or understand polygenic risk scoring systems, then I encourage you to start kind of building your toolkit surrounding molecular epidemiology early. And in medicine, all of this is very important. Understanding genetics as it pertains to even cardiovascular disease is very complex. And so, if you're looking to go into a specialization like endocrinology or cardiovascular, or even trying to get into research in general, this is a good place to be. Um, so then the authors go into some of these very essential plots, the Manhattan plot and the QQ plot, to visualize GWAS results. So the Manhattan plot shows significance of each variance association with a phenotype. So in this case, the phenotype that they were looking for was body mass index. And so each dot represented a single nucleotide polymorphism, SNP, ordered on the x-axis, and then according to their genomic position, y-axis. Um, so according to that, that represents kind of the length. Um, and so these associations you can kind of map out in these wonderful plots. And then there's Bayesian models that you could be exploring as well. Just for the sake of completeness, this has been incorporated. Um, So now we try to end with types of applications and risk prediction. Polygenic risk scores are commonly used to predict the risk of disease in a target cohort. So using the GWAS summary statistics. Um, Polygenic risk scores, they can be used to identify individuals that are high risk in disease, and that's the idea. Like in prevention, you want to discover risk scores, the risk of disease ahead of time, so that you can prevent certain segments of the population, prevent disease in certain segments of population. That's why disaggregated data is so important. So again, risk prediction is important in the context of GWAS. And using the summary statistics, of course, this is important. But they're weighted sum scores of risk alleles. That's what they are. And so different SNPs have different effect sizes. And so understanding the effect of not only phenotype, but also the polygenic risk score, and how much various genes play a role or contribute to that whole overall score of disease development, that varies. So as you can see that in the polygenic risk score distribution, individual one, if you looked at it within that bell-shaped curve, individual one fell way differently than 2, 3, and 4. So this is part of that targeted therapy. Understanding even social determinants of health in terms of polygenic risk score is probably a lot more informative than looking at only phenotypic expressions as well. Um, and so, for instance, one of the best performing PRSs, polygenic risk scores to date, has been developed for glaucoma. Individuals in the top decile of the score distribution have a 4.2-fold increase in risk compared with the bottom 90%. So, a commonly used metric for assessing polygenic risk score accuracy is the area under the receiver operating characteristic curve. And this is one that I have lectured on, so please refer to this lecture when understanding this curve. But it's the performance of the models and the ability to discriminate between two groups. And the best performing model, a threshold must be set at which, to classify individuals as high risk. Choosing a threshold is based on weighing the costs and benefits of false positive versus false negatives. Um, so, of course, AUC curves are important in diagnostics. Um, so a high AUC or odds ratio even does not promise an enrichment of high risk individuals in the top percentile of the score distribution. Um, so there's a reclassification index, which may be more clinically relevant. And so there's this whole discussion on the effect of GWAS and potentially being useful um, in diagnosing certain providing risk categorization for certain diseases. And for glaucoma, that has been the case. The risk categorization has been extremely effective. But this all has to do with the quality of the data that is in existence, and the number of data. And then there's a whole discussion on data equity, collaboration, and data sharing. Um, but the GWAS catalog is a good place to be, because they go into some of the summary statistics, and some of the lead SNPs are reported in GWAS papers. Um, the UK Biobank GWAS summary statistics, another good place. Gene Atlas, so GWAS catalog. We can just quickly click on GWAS catalog. Here we are. And you can download a full copy of the GWAS catalog in spreadsheet form or summary statistics. Um, you can submit summary statistics to GWAS catalog. Um, so you can do a quick search kind of, and um, take a look at the number of studies. Breast carcinoma, 159 studies. Associations, 100 and 2000, about 2,000. Um, Traits, 16 traits identified. I know part of the screen you can't really see, but 16, and then one gene related. So these are all trait related articles. I can already tell because it's T, labeled as T. And so you can kind of take a look at HER2 positive breast carcinoma. Let's take a look quickly at the types of synonyms that are available. And you can see HER2 receptor, HER2 positive, HER2. So these are the types of terms that you might be able to take a look at as it pertains to that. And then there's 3 reported traits. And so that's how you can kind of take one topic and then build on that. So there's a total, actually, in this catalog, there's a total of 209,000 SNPs in place, approximately. So that's the GWAS catalog. Gene Atlas is another one. Again, a good place. Pan UK BBGWAS Atlas collection of publicly available GWAS summary statistics with follow-up in silico analysis. FinGen results. DBGAP, also public depository of NIH-funded genomics data. And here is, and I'm going to be probably taping a separate recording on this one. This is just so important. But this is the database of genotypes and phenotypes, and which is developed to archive and distribute the data and results from studies that have investigated the interaction of genotype and phenotype in humans. So, for instance, Gabriella Miller, Kids First Pediatric Research Project in Microtia in Hispanic populations. Yeah, I think I lectured on this already, so I'm not going to spend too much time on this. But just know that this is available. OpenGWAS, again, OpenGWAS database is a good place for exploring data sets. This is a place where there's a lot of data sets available from 42,334 GWAS summary data sets are available. So if you want to take a look at fine mapping co-localization, then you can use GWAS glue for that. You can alternatively kind of start by taking a look at the number of SNPs as it pertains to certain disease conditions, and kind of go from there. So this is another useful resource available that is open source. Again, data sets, secondary data sets, very important secondary data sets, which allows for proper dissertation writing if you're looking into that, or whatever it is. If you have research that you're trying to share, collaborate, um, take a look at this website. Open. Um, this is OSF, and this is about open science workflow. So, kind of sharing your workflow with other people, and that is just as important as the actual finished product itself sometimes. So it's an open science framework. That's OSF stand for. That's about that's about it. I'm not gonna make this way too long, so much so that you can't really make sense of what's in here. But there's so much to include that I couldn't help but click on some of these things. And International Common Disease Alliance. Let me move this a little bit. So this is about biobanks. And so, yeah, so this is another place where you can kind of take a look at common diseases and how biological mechanisms, along with social factors, are working together to cause disease. So that sums up everything that needed to be talked about here. The bottom line here is that just go ahead and explore. Explore all of these resources, everything that's included within this nature review. Just explore and experiment, and the more you experiment, the more you'll learn. That's how research works. Thank you for listening.